Okay, we're going to talk about the control of blood glucose concentration in the blood. You can see here that the normal range is between 70 and 130 milligrams per 100 centimeters cubed blood. If you're fasting and you don't suffer from diabetes, then the blood glucose level is around about 100 milligrams. Of course, that will go up briefly after a meal, and it will fluctuate a little bit, but it will remain about that level. Now, why do we need to control blood glucose concentration? Obviously, if it's too low, then there's no respiratory substrate. There's no glucose. The glucose is the main substance which enters glycolysis, which then goes on to citric acid cycle and oxidative phosphorylation to make ATP. So not enough respiratory substrate. So the cells won't be able to function properly. And of course, if a person has very low blood sugar, such as they've taken too much, diabetic person who's taken too much insulin, then they will fall into a coma, as there's no respiratory substrate for your nervous system, for your brain, and so it can't function properly. Now, why is blood sugar, when it's too high, why is that a problem? Okay, well, when it's too high, glucose, of course, let's say it is osmotically active. And what we mean by that is it tends to draw water. If you've got high blood glucose, it will draw water into the blood, increase the blood volume. That will lead to a high blood pressure, and over time that causes damage to the heart and to the kidneys and to other organs as well. So it's got to be kept within this range. Okay. Now, the organ which is principally involved in controlling blood glucose is the pancreas, okay, which of course has an exocrine function as well. It secretes digestive enzymes, but it's got an endocrine function. It makes two hormones, glucagon and glucagon. They're both peptide hormones. Glucagon will tend to raise blood glucose. It does lots of different things, but its overall effect is to raise the blood glucose. So at times of starvation, your pancreas makes glucagon, and it has that effect. Insulin, that does the opposite of that. That will tend to lower blood glucose. All right, let's talk about glucagon first of all. Well, before we do that, we need to just have a little think about this molecule here, which is, you may recognize it as glycogen. Okay, so we've got glycogen there, which is a polymer of glucose. So, and you can see here, you've got a glycosidic linkage between carbon atom number 1 and carbon atom number 4 of glucose. Here's glucose, over here, that's the monomer. So glycogen is a polymer of glucose, and it's found in large quantities. Principally, you find it in the liver and in the skeletal muscle. So the two main target organs of glucagon here, glucagon affects mostly the liver and the skeletal muscle. Okay. Now, what happens if there's a glucagon circulating in your blood? That means your blood glucose level is a bit too low, and you need to convert some of this glycogen here. You need to convert it into glucose. So you can see glycogen is made up of N residues of glucose, and this enzyme glycogen phosphorylase here, what that does is, right, you use a phosphate group, an inorganic phosphate group just drawn with a P with a circle around it, but don't forget that is actually really it's like that. It's PO43-, that's the phosphate group. So that will cleave off a glucose from this end. Okay, so what happens here is the phosphate group is going to stick onto that carbon there to form glucose 1 phosphate. Okay, so it will be on carbon atom number 1, which we've got over there. That's carbon atom number 1. And then what's left behind is you get a shortened glycogen chain, N-1, and that's ready for another one to be cleaved off by the enzyme in the next step. So that's what happens during a period of starvation. Glycogen, the enzyme glycogen phosphorylase is actually switched on by glucagon. That makes it active in the liver, and it will reduce glucose 1 phosphate. As I say, that can happen in the liver and in the skeletal muscle. And that's what the process is the same there, but after that it's a little bit different, because if you think about what here, right now the liver is the glucose 1 phosphate is then converted into glucose 6 phosphate. That's just an isomerization reaction with an enzyme that does that. Present in the liver, and it's also present, that enzyme is also present in the skeletal muscle, so you get the glucose 1 phosphate going to glucose 6 phosphate. That's where you get a bit of the difference next between the liver and the skeletal muscle. In the skeletal muscle, that glucose 6 phosphate is then just used for glycolysis, because of course skeletal muscle has a very high energy requirement, particularly during exercise, obviously. And so all that glycogen will be converted into glucose 6 phosphate and directly into glycolysis. In the liver, it can enter glycolysis as well, but in the liver, there's also another enzyme present, which is not present in skeletal muscle, and it's called glucose phosphatase. And what glucose phosphatase does, it cleaves off the phosphate group from carbon atom number 6, which is that one up there. There's carbon atom number 6, chops it off and turns it into glucose. And this glucose can then leave the liver cell into the blood. So it raises blood glucose in that way. And then that glucose, once it's in circulation, can be used by all the other cells in the body. Whereas skeletal muscle, that doesn't want to provide the rest of the body with glucose, because it's got a very high glucose demand itself. So all of the glucose made there stays as glucose 6 phosphate. If it's glucose 6 phosphate, it can't pass through the cell. It can't leave the cell. Once you lose the phosphate group, that can leave the cell. It passes out through a glucose transporter, which through diffusion, it's not active transport, doesn't require ATP, it's just a proteinaceous channel which allows glucose to pass out of the cell. Okay, so glycogen, breakdown of glycogen occurs, glycogenolysis, okay, that occurs. However, if starvation continues and the glycogen is all exhausted, then obviously you can't make any more glucose that way, but you need glucose. So what happens then is this process, which is called gluconeogenesis. So the clue is in the name. Neo is new, genesis is making, glucose obviously. So making new glucose. New glucose is made from some of these precursors here. So lactate, if there's any of that about from anaerobic respiration. This, by the way, I should say that gluconeogenesis mostly occurs primarily in the liver. I think it occurs a little bit in the kidneys as well, but most of the liver is the main site of gluconeogenesis. It doesn't happen in skeletal muscle. So gluconeogenesis is switched on in the liver cells by glucagon, and that happens when all of the glycogen is depleted. Some amino acids, so you hear glutamate, aspartate, serine, and alanine, they can be used to make glucose. So the amino group is chopped off, and then the carbon skeleton is recycled into glucose. Glycerol also, which is of course part of triacyl glycerides, fats, that can also be used as a precursor to make glucose from that. Right. So that's what happens during the periods of starvation. Let's look at the opposite of that is when you just had a meal, and the beta cells of the pancreas will be, and we can talk about how they detect low levels of glucose. We'll talk about that in a later video, but they make insulin. And insulin affects not just the liver and the skeletal muscle, it's practically every cell in the body has insulin receptors. Now what it does is, once the cell gets insulin bound to the surface receptors, it will cause this protein which is called GLUT4. It's a membrane channel protein which allows glucose to pass into the cell. Now, I've drawn a little diagram of it here. You can see here I've drawn these. This is supposed to be vesicles. Can you see that there? That's a vesicle. A little membrane, bit of membrane inside the cell, enclosed part of the endoplasmic reticulum, and the little red marks in it, that's supposed to be the GLUT4 protein. Okay, so those little red bits, that's GLUT4, and they're not normally on the surface of the cell, but once insulin binds, so here you get insulin, that will cause those GLUT4 to be transported onto the cell membrane. You can see that it fuses with the cell membrane there, and now you've got loads of those GLUT4 proteins on the surface, and they are membrane channels which allow glucose to enter the cell. Again, it's by diffusion, it's not no ATP, it's not active transport. So that's one of the main effects of glucose, is to let glucose into cells. The cells, glucose can't get in without insulin. Also, what they do to the cell is they do the exact opposite of glucagon. They tend to promote glycogen synthesis. It promotes glycogen synthesis here from glucose. So the glucose which does enter the cell will then will make glycogen. That's in the liver and the skeletal muscle mainly where you get glycogen, of course, not really in other tissues. And it will also cause fatty acid synthesis. Okay, so, and that's particularly important in adipose tissue, in fat tissue. So the glucose will be broken down, if you remember, to pyruvate, and then it will go, first of all, it's broken down to pyruvate, and then it's broken down to acetyl-CoA, which is in the link reaction, and acetyl-CoA, there's 2 carbon units, they're built up to make fatty acids. And the fatty acids then go to make triglycerides, which are stored in the adipose cells. So glucagon, as we say, lowers, tends to increase blood glucose during periods of starvation. Insulin, on the other hand, does the exact opposite. It tends to lower glucose concentration after a meal.